everybody. Today's lesson is going to be on blood. We're going to look at it from a scientific standpoint, and then later we'll relate it to more issues of forensic science. So our learning targets for today: be able to describe the composition of blood. What's it made out of? Number 2, explain blood's function. What does blood actually do? What's its job? Number 3, describe the structure and function of a molecule that we call hemoglobin. And number 4, explain how and why blood cells are made and why they're destroyed. Number 5, describe the function of each type of substance or cell that we find in blood. Let's get to it. We're going to start off with the composition of blood, or what blood is actually made of. There's 2 major parts. One is going to be the plasma, the liquid portion of the blood, and the other one are the formed elements, or the solid things that are found within that liquid portion. Now, our formed elements are going to be broken down even further into erythrocytes, leukocytes, and platelets. Now, erythrocytes and leukocytes, if you translate their names, what they basically break down to, erythrocyte means red and cytes means cells. So erythrocytes are red blood cells. Sometimes we nickname them RBCs. Leukocytes, leuko, the prefix means white, cytes means cells once again, so those are our white blood cells, or our WBCs. Now, platelets, they're tiny little proteins that are going to help clot wounds when we're injured. So they kind of patrol the bloodstream, and when there's an injury, they're going to basically migrate towards that injury site, and they're going to essentially do the job of plugging up a wound if there is an open wound in the body. So how we get information about the composition of blood is with what we call a centrifuge. A centrifuge is a machine that spins blood really fast in order to separate the components. So if we look down in the picture here, we can see number 1, withdraw blood and place in tube. We see it then put in a machine that seems to be spinning very fast. This is called centrifugal force. And what it does is it forces very heavy substances to the bottom of a tube, whereas the lighter substances will rise to the top. So once you're done with the centrifugation process, you can see the components of blood. About 45% of the blood is your red blood cells. We can see that less than 1% of it here is this white layer called a buffy coat, which is made of leukocytes and platelets, and all 3 of these make up our formed elements. So the remainder of blood then is our plasma. It's this yellow straw-colored fluid here. About 55% of the composition is plasma. It's a watery-like liquid that all those formed elements actually are suspended and float through. The main function of blood is going to be transport. We're going to be transporting a lot of substances throughout the body. Everything from oxygen and carbon dioxide and glucose and hormones. The list goes on and on, but main function is definitely transport. Another function that blood has is to regulate heat within the body. Now, we know that the human body operates within a very narrow range of temperatures. We are centered around 98.6 Fahrenheit or 37 degrees Celsius. And the body does not do very well when it goes much higher, much lower than that. So what helps us with that is the fact that watery fluids like the plasma and the blood aid in temperature regulation. Water has what we know as a very high specific heat, the highest of all substances. It takes a lot of energy to change the temperature of a large mass of water, either up or down. So part of blood's function is to make sure the temperature of the body doesn't go too high or too low, which is bad for the organism or us. Let's take a look at the breakdown of blood within the body. You can see that it comprises about only 8% of our body weight, where all the other fluids and tissues are up to about 92%, the difference. And this is kind of surprising to a lot of people. Most younger students think that blood absolutely just fills your body from the bottom to the top. And in reality, all the blood that you have is enclosed in a set of tubes, and there's not as much blood in the body as people think. And we'll get to that in a later slide. The blood that we do have, the 8%, we already mentioned that 55%, over half of it's plasma, and less than half of it would be those formed elements. If we take a closer look at what's inside the plasma, notice that water is 92% of that plasma. Proteins that float within the blood are about 6%, and other solutes such as ions, nutrients, waste products, gases, regulatory substance, they only comprise about 2% of the remaining. Now, as far as our formed elements go, within that 45% of the blood, erythrocytes, as you can see, 4.2 to 5.8 million per milliliter, leukocytes, 5 to 9,000 of them per milliliter, and then platelets, about 250 to 400,000. They're very small in comparison. The leukocytes further break down, as you can see, into a number of different types, and we'll cover those a little bit later in lecture as well. How much blood do we actually have in our body? What is our blood volume? For most adults, it's around 4 to 6 liters, depending on the body size. And we usually talk about units of blood when we talk about blood donations. Units of blood are about a pint. So if we go ahead and we kind of translate into what 4 to 6 liters is and how many pints that is, we can say that about 8 to 12 of the bags that we donate here, or pints of blood, is typically what you have running throughout your circulatory system. Imagine you had 2 liters of pop, and that would be about the equivalent to how much blood you have inside of your circulatory system of your body. Now, we can donate a pint of blood without any problem at all. Our body will sense that we've lost some blood, and it will work to go ahead and create more to replenish the volume that we donated. Now, when injuries happen and blood is lost in great quantities, what we can start to see happen is different things depending on how much is lost. It can go from a woozy feeling to a person losing consciousness. And once you've lost about 20% of your blood volume, a person could go unconscious at that point. Once you lose even more, up to about 40%, then we're riding a thin line where someone may or may not make it based on that amount of blood loss. We're going to focus in on those formed elements at this time. First, let's talk about erythrocytes. Again, those are your red blood cells, or RBCs. These cells are unique in that they're unlike the cells that we study in biology, in that they don't have a nucleus anymore. In fact, through their development, they lose their nucleus. And their shape is a little bit different. Now we have, instead of kind of a round, blobby, spherical type of cell, we have a tiny biconcave disc. So there's kind of like this little depression in the center on both sides. Red blood cells do not contain ribosomes, mitochondria, other organelles typical of most body cells. So the endoplasmic reticulum, the Golgi apparatus, lysosomes, all those things. This RBC is very minimal in terms of what's inside of it, but it is the most numerous of the formed elements. In fact, there's 5 million red blood cells per single milliliter of blood. One of the most important components inside of a red blood cell is the molecule hemoglobin. And this molecule's job is to basically carry the oxygen that gives all of our cells the oxygen it needs to stay alive and carry out the jobs it does. And it also can carry the waste gas, carbon dioxide, which we need to unload regularly through exhalation when we breathe. Let's dive a little bit deeper into the protein hemoglobin. Within each red blood cell, there's approximately 200 to 300 million molecules of this protein called hemoglobin. It's made up of 4 globin chains, which you can see here. We have alpha 1, alpha 2, beta 1, and beta 2. And these 4 chains together have what's known as a heme group in the center of each one. And it's this heme group that actually allows us to bind oxygen. And when we do bind that oxygen within the lung tissue, we then go from hemoglobin to the molecule oxyhemoglobin. And this molecule that is now loaded up with oxygens is ready to deliver it wherever oxygen is needed, from the top of your head to the tips of your toes. It's going to deliver that oxygen to cells that need it. So where do red blood cells come from, and where do they go? So we have this process, it's called erythropoiesis, and that is officially the name we give to the process where red blood cells are made and formed. It begins in your bone marrow, in the hollow spaces within your bones, and they begin from cells called hemopoietic stem cells. So these stem cells don't really have jobs or identities yet, but what they're going to do is they're going to go through a whole bunch of stages of development to ultimately, at some point become these erythrocytes. It takes about 4 days for this process to occur. And we need it to occur all the time because number one, red blood cells have a very limited lifespan, which we'll talk about in a second. And number two, they are also created and destroyed at an incredible rate of 100 million per minute in an adult. So that's where they come from. Ultimately, they're going to meet their end after about 105 to 120 days, so about 4 months. Once they get worn out, we have these white blood cells called macrophages that are going to go ahead and recycle aged, abnormal, or fragmented RBCs that just aren't capable of doing their jobs well anymore. And when they do, they're going to break down all the products of that red blood cell into amino acids, iron, and a chemical called bilirubin. Let's go over a process where there's a definite need for red blood cells to be made aside from the normal birth and death rate of them. Let's say that you and your family decide to take a vacation somewhere up in the mountains to high altitude. As you can see at the top of the picture here, higher altitude is linked to having less atmospheric oxygen present. Now, your body will sense that, especially if you've lived your life closer to sea level. So ultimately, it's going to be a little bit harder to breathe there. Your body is going to go through some processes where it identifies that there is lower oxygen in the air that you're normally used to. So what will happen here is your blood oxygen will start decreasing. Now, you have a mechanism of detection, and it's going to be found in your kidney organs. So it's going to, as you can see, there's an actual value in the middle. However, the value of the oxygen is going to go down. Your kidneys can detect that. And what it's going to do when it departs from that set point value is it's going to release a hormone within the blood called erythropoietin. You can see it right here. It's these little blue molecules that you see coming out from the downstream of that kidney. Now, that little molecule is going to travel everywhere within the body, and one of the places that it will also be detected is in that red bone marrow that we discussed. The red bone marrow will then send its own signal to start producing greater amounts of precursor stem cells, which will turn into red blood cells through that 4-day development that we mentioned. So you can see it will go through a number of stages. Here's a hemocytoblast, goes to then some RBC precursor cells. You can see it goes through a number of changes and eventually leads to more red blood cells. And they'll start flooding into your bloodstream to kind of alleviate what you've lost through the decrease in oxygen that you're normally used to. So in other words, your body will compensate by producing more red blood cells that can carry the oxygen that's present in the levels that you find in that new environment. This will follow multiple systems in your body called feedback loops. So blood formation will follow a homeostatic mechanism called negative feedback. Homeostasis was when your body tries to maintain a normal range of values for a whole bunch of processes that happen inside of it. One of them being your oxygen balance and oxygen and carbon dioxide balance. So as the oxygen goes down, your body has a mechanism for building the amount back up and letting your body go through the processes that it's normally fully oxygenated for. So mechanisms operate to balance the number of cells formed against the number of cells destroyed. When erythropoietin is produced in the kidneys, it makes a signal to make more red blood cells, and it happens when these organs sense low oxygen or O2 levels. When a red blood cell has reached the end of the line, there are macrophages in your spleen and your liver, which are going to go ahead and they're going to take apart these aged, abnormal, or damaged erythrocytes and break them into 3 components. One is the protein globin chains that we mentioned earlier in hemoglobin, and those can basically be broken down into their separate amino acids, which ultimately all proteins are made of. There is iron inside the heme molecule, which can then be utilized and reused within the bone marrow to help build new red blood cells. And there's a pigment called bilirubin, which winds up getting filtered through the liver and excreted into your intestine as part of bile. Bile will then essentially be absorbed by fecal matter and then released from the body. This is a diagram of those uncommitted stem cells that we find in the bone marrow. And eventually they're going to give rise to committed cells. So these cells right here, these brand new cells, can become different things within the body. One of the routes they could take is down this left-hand column here, where you begin as a pro-erythroblast. Your DNA has kicked in. It gives you the job role that you need based on the needs of your body, and it will go through a number of stages to eventually become that erythrocyte, that red blood cell. We also have this middle column that goes really from left to right over here, and these are all your white blood cells. So monoblasts will become the white blood cells called monocytes, lymphoblasts become lymphocytes, and then myeloblasts can become neutrophils, basophils, or eosinophils, depending on the needs of the body. And these all work for your immune system and help keep your body safe and healthy. The last route one of these stem cells could take is through the megakaryoblast route. Eventually, these produce very large cells that pump out those platelets that we were talking about that plug up wounds. So our white blood cells, which we called leukocytes earlier, their function then is immunity. And if you take a look at the picture here on this slide, you can see different types of white blood cells. Here's a monocyte, a lymphocyte, neutrophils, eosinophils, basophils, and all of these cells, again, they're going to do their own special job and fight off pathogens or things that are making you sick, depending on the type that have infected you. So you'll have different response rates from different types of white blood cells, which can be looked at and then interpreted into what potential infection a person may have. There's not a lot of white blood cells in comparison to red blood cells. In fact, 1 milliliter, where it contains 5 million red blood cells, only contains about 5,000 to 9,000 leukocytes, with different percentages for each type. These cells, however, do have their nuclei intact. They're fully functional cells like we study in basic biology. And again, they have clinical significance because they change with certain abnormal conditions. Now, in forensic science, when we take blood samples, it's typically the DNA from white blood cells that we're able to sample and then run through the process of investigating the DNA in it as we look for matches with that of other people or crime scenes. Platelets are ultimately little acellular bodies whose function is clotting. So they circulate around the blood. They look like pale bodies when we have them underneath a scope. And they're much smaller than red blood cells as well, about half the size of a red blood cell. They do have some important properties within their job set. Adhesiveness is a measure of how sticky they are to a particular tissue. And aggregation, as well as agglutination, will refer to their ability to clump up together and aggregate in high numbers in order to plug up a wound, where the outside environment is allowing germs into the inside environment. The numbers are pretty high for these per milliliter of blood, about 250,000 per single milliliter. In this picture here, you can see that we're kind of looking at the skin from the inside out, and you can see that there's been a breach in the skin right here, some kind of cut, and we have potential pathogens in this area, and all these little kind of spindly purple bodies here, these are the platelets responding to it, and they're going to come together, they're going to stick together in high numbers and start plugging up this wound. When you are injured, like you see here in this picture, the cells that are injured are going to put out kind of like a chemical alarm. And when they do that, they're going to activate the platelets to respond and create a fibrin mesh over that injured site, kind of like a little biological band-aid. And what that does, it provides a scaffolding for tissue repair. So red and white blood cells are also going to congregate to the injury site. The red blood cells, as they're exposed to the air, are going to basically turn into a scab that someone would get on their skin once that skin is breached. And the white blood cells will also respond to that area in order to clean up and eat any bacteria or pathogens that have entered in that area. If they do get caught up within the clot themselves and they are battling an active pathogen, what we may see is the formation of pus at that wound, which is kind of a white liquidy substance that is full of white blood cells. Let's take a look at this complicated set of events that occurs when someone is injured. Here we can see a person has stepped on a nail. There's an injury. Obviously, pain is going to come with an injury like that. And the cells that are affected are going to run through what we call a chemical cascade in order to turn on all the switches to plug that wound. So you can see we have something called prothrombin, which when in combination with prothrombin activator and calcium, will turn into eventually the active chemical thrombin. Thrombin with another chemical called fibrinogen will turn on the production of the protein called fibrin, which will ultimately produce the scaffolding or the meshwork that all the red blood cells, the platelets, the white blood cells can get trapped in, but will plug that wound and eventually get you on your way to healing where that breach of your body happened. We've made it to the end of today's lecture. Let's reflect back on our learning targets. We are to be able to, number one, describe the composition of blood. What was blood made of again? Number two, what is blood's function? What's its main job? Number three, what is the structure and function of hemoglobin? What's it for? How is it put together within each red blood cell? Number four, explain how and why blood cells are made and destroyed. And number five, describe the function of each type of substance or cell in blood. Okay? Take some time, watch the lecture over if you need to, pause it when necessary, and we'll see you next time. Take care.